Welcome, everyone. My name is David Rivers. I am a professor of biology at Loyola University Maryland. I'm trained as a forensic entomologist, and I have this unique trait of studying flies during their times of spitting and puking. And I'm going to spend time actually trying to present to you this afternoon about fly pattern evidence in which insects themselves actually create contamination at crime scenes. And it's a relatively understudied area that I think is fascinating and one that actually needs more attention. So the plan is to introduce you to the topic, show you some pictures, do a little bit of show and tell, and give you a different perspective of forensic entomology. In fact, from that regard, I am a forensic entomologist, and I think that most people understand that forensic entomology is usually viewed from a positive standpoint for criminal investigations. From the standpoint that the most common use of insects has been that the development of bugs on bodies, as I like to say, can be useful for estimations of portions of the postmortem interval. There are many examples in which that has been useful for a death investigation, and there are many other uses for insects that are considered to be aiding a criminal investigation. What has received much less attention, however, is when insects actually contaminate crime scenes. And I'm talking specifically about flies and adult flies in particular. The reality is that when insects are attracted to a death scene, it's because they're there to dine. They are interested in foraging upon either the corpse or the body fluids that have actually been released, particularly if we're talking about a violent crime. And they're there to eat on any of those types of fluids. And to be honest, they're very, very sloppy. And they can actually do a number of things that are actually not of benefit to the criminal investigation. In fact, I like to think about the association between flies and evidence at the scene as having the potential where the flies can modify the existing evidence that actually exists. So if we think about blood stains in particular, then flies are already known to modify the shapes of some of the blood stains that are actually found at a crime scene, which is not aiding in the reconstruction efforts. And in fact, they can go so far as to destroy the evidence as well through their feeding activity. This is something that we certainly understand that they do when the adult fly feeds upon the corpse and or deposits eggs or larvae. They start to consume the largest physical piece of evidence that's actually associated with a death investigation, the corpse itself, but they can consume entire stains at the same time, leaving little or no evidence of the stain itself. But then to counter those two relatively negative aspects, flies do have the ability to preserve some forms of evidence, at least for a temporary period of time. And what I mean by that is that the fly can actually feed upon a body fluid, and again, let's say blood, and then when they do, they have a tendency after that they've had it in their digestive system for a period of time of either regurgitating or defecating and creating a new stain that might be in some other location from where the corpse was originally. And those stains will dry very, very quickly. So in essence, whatever was in the stain, meaning the proteins and the DNA and sometimes drugs that were in the tissues that they fed upon, are now locked in a dry state and preserved that might be valuable evidence at a later time, particularly if the crime scene's already been cleaned up and someone discovers later that there are actually these fly stains that are actually located somewhere else, such as around a window or on a lampshade or some other artificial light source. So that we have different potential outcomes that can be the result of adult flies actually showing up to a crime scene. And we can see that the majority of the examples I've given you so far are the flies doing something destructive that's not aiding in the investigation. And that's going to be our primary focus here. This is the pattern evidence that the flies are actually creating. Other insects do as well, but flies tend to be the most abundant of the types of insects that will actually show up under most situations. So they will be our primary focus over the next few minutes when we're talking about pattern evidence created by insects. Now, again, as we talk about what adult flies can actually do, there tends to be two broad categories that we think about when we associate insects interacting or associating with any type of stains that might result from the deceased individual. One is the modification that I've already referred to, and the reality is flies will interact with wet or dry stains. When they do, however, there are differential outcomes that come about, and that will be evident as we actually look at a few examples in a few minutes. The other thing that can happen is that the flies, through their feeding activity, can create unique stains. That can also be as they walk through wet blood and then deposited some type of transfer pattern, and they come in more than one form. And again, we'll take a look at these in a moment. So that's more the modification of the stain. It's also creating new stains. And through their normal activities of their digestive tract, again, this idea that they actually will consume the food, and in some cases actually enter into bubbling behavior that we can see in the upper right-hand corner here, where a drop of blood is dangling from their mouth parts. When they drop that stain at a crime scene, then they've created a new stain that can have the appearance in many ways to certain forms of blood stains. And the same can happen when we're talking about fecal elimination with these flies, because their feces is liquid, and oftentimes that feces is going to have the same color as the food source that they've consumed. Again, we're referring to the body fluids at the crime scene, and those stains may be confused at times or very difficult to distinguish between certain types of blood stains and other body fluids depending upon the context. Now, this type of association has long been recognized by individuals that are working crime scenes, particularly bloodstain analysts, who've actually coined the term insect stain in reference to bloodstains that are created or resulting from insect activity. Now, this definition does not actually specify any particular type of insect, nor does it reference any specific type of mechanism. So it's a very broad term from the standpoint of forensic entomology, but in most cases we tend to be thinking about flies are the culprits in this particular case, and we'll see many examples. This stain or this term also is very specific from the standpoint it's only referring to modification of blood stains, and we know that flies are not particular in the food that they will actually feed upon. They're seeking foods high in protein. And the reality is they will actually feed on a multitude of other types of materials. So not only will they feed on a multitude of materials, they will feed and create a wide range of other types of stains that go well beyond just blood stains. In fact, in many cases, depending upon the fly species and the context, we've been able to identify a large number of different types of stains that are being created by different mechanisms. We will look at many of these as we proceed through this talk this morning, but at the same time, there will be some that will be specific to certain circumstances that are not going to be associated with blood. So if it's not associated with blood, we really won't make it much of an emphasis for right now. But the point is there's a lot more types of stains that are actually produced than what most people actually think, and that's been reported in the literature. And that has led to coining a broader term than just insect stains. So insect artifacts really kind of encapsulates this idea that flies feed upon more than blood. They modify more than just blood at a crime scene. And there's a wide range of stains that are actually being created. So by mechanisms that have not been addressed necessarily widely in the literature as well. In fact, you can think of insect stains being a subset of the term of insect artifacts. And I'll have a tendency to move back and forth between these terms. Since we are going to focus on blood throughout this talk, then we really are talking about insect stains by and large, but you're going to see that if you have some familiarity with this topic, it's going to change your perspective, I think, just a little bit. Regardless of how that we want to define it, there are some basic characteristics that we can actually talk about with insect artifacts. One of those is that the morphology of the stain itself tends to be a reflection of the surface material of which it's been deposited. Now, what that means broadly is that the topography and the porosity of the surface material is going to influence the appearance of the stain, but so will the surface chemistry. And there will be unique interactions that take place. And I'll try to describe those as we actually go. We will see that most evidently when we are actually making a distinction between stains that are deposited on non-porous surfaces versus those that are deposited on textiles and fabrics. And in the grand scheme of things, even the information that's associated with human blood stains on textiles and fabrics is minuscule by comparison to the information that's out there on non-porous surfaces. Color of insect stains, insect artifacts. I think that there's examples in the literature that refers to insect stains being relatively easy to distinguish from human blood stains based upon color characteristics. That is a statement that can only be made within context, because it is dependent upon not only the surface material and the degree of contrast or the background color of the material that's been deposited, but there are most definitely species differences that actually exist as well. And it would take too long to try to give you every single example during this particular talk of the variation. We've looked at 37 different species of flies, so we know there's a lot of variation that actually exists. There is literature that indicates that you might be able to have specific characteristics in terms of alternate lighting with various types of insect artifacts, fly artifacts in particular. The literature is still in its infancy. There has been some work that's been done recently that suggests you might be able to distinguish any type of insect stain from true blood stains, but all of that work so far has been done in a laboratory setting. So we will avoid going too far with it, other than to simply say there's the possibility that you can use alternate lighting, because digestive artifacts, those that are created from the digestive tract of the fly, tend to be unique from the other types of artifacts that we can look at. So with that in mind, we're getting geared up so that we can start doing some show and tell. So that I can introduce the types of stains to you. What I want to do first, though, is just give you just a brief synopsis of all the stains that we have observed at this point that could potentially be produced by flies. There are 4 that stand out as the most common that you would encounter when the fly has associated with blood. Those 4 wind up being regurgitate stains, defecatory stains, translocation, and tarsal tracks. The first 2, regurgitate and defecatory, I commonly refer to as digestive stains. They're produced from either end of the digestive tract. They are by far the most common ones that we would actually see at a crime scene. And they have unique morphologies and they have unique mechanisms that lead to that. The latter 2 are far less common, but under certain conditions and with certain flies, we tend to see translocation more frequently than we do in other situations. Tarsal tracks, for example, or what we refer to as fly footprints. The fly has walked through blood, or it's walked over the top of blood, and actually then transfers footprints, fly footprints, to a non-bloodied surface, are very common with smaller bodied flies, particularly in the genus Chrysomya and in Lucilia. Much less common when we're actually talking about a flesh fly, a sarcophagid, or some of the larger bodied blowflies, such as Cinnamya, or some of the larger califorous species in particular. In terms of how are these stains produced, regurgitation is actually the result of expulsion of food material that's actually in the foregut of the adult fly's digestive tract. So this means from the crop forward, that food is actually driven forward through the mouth parts, and usually it is dangling from the tip of the mouth, and is most commonly observed as bubbling behavior. And there's a number of working ideas behind why do flies actually bubble. What we do know is that most species that would be associated with the crime scene will drop that bubble or the regurgitate at the crime scene, either intermixed with other stains or in unique locations. And the full intent is for the adult fly to come back later and feed upon it. In fact, our observations suggest that flies are far more interested in feeding upon regurgitate that contains blood than actually feeding upon human blood itself. This regurgitate is not to be confused with vomiting or vomitus, which was actually reported in some of the early literature in the 1970s and 1980s. Vomiting is an entirely different mechanism that takes place with adult flies. And actually, the contents are actually derived from the midgut, not from the foregut. Now, this may not seem like of importance to crime scene investigation, but it is from the standpoint that the vomitus is actually chemically distinct from regurgitate, and it also has a different morphology as well. Flies have a tendency not to engage in vomiting on a frequent basis, so you're not expecting that vomit would actually be associated or any type of vomit stains with the crime scene under normal situations. The other stain that's very, very common are the result of fecal elimination, defecation, which would seem like it should be just a very straightforward to say this is how the flies actually defecate, but there's really not much that is normal nor simple when we're talking about forensic insects. And in the case of flies, they have at least 3 mechanisms that they actually use for eliminating feces. One is that the fly is stationary, and the body is positioned more or less parallel to the substrate that it's resting upon, so that when it forces waste material out of the anus in this case, the drop actually will be pushed out passively, and it falls more or less perpendicular to the surface at which it's deposited, and also a very short distance. The net effect is that the resulting stain will generally be round or circular in appearance, with no stains and no evidence of edges such as spikes or scallops that you might see with certain types of blood stains that have fallen perpendicular. In contrast, if the fly is actually stationary but then begins to move, meaning to walk, to run, or even to fly before it has completed fecal elimination, there will be a tail that will be associated with that fecal stain that will taper in the direction that the fly was actually moving. In some cases, that tail will be very long, that suggests that the fly was walking. In other cases, if the fly is running, sometimes it's tapping its abdomen as it's doing so, creating almost a dotted line appearance to the stain itself. And then finally, the third mechanism that they can use is that some flies, not all that we've actually observed, have the ability to protrude the anus and forcibly expel liquid feces parallel to the surface at which it's resting upon, in which case that the feces is airborne and has been pushed out of the anus under high pressure, so that the characteristics of that airborne liquid are somewhat similar to certain forms of blood spatter. So when the feces actually then makes contact with the surface, the drop will still continue to move, creating a tail that's pointing in the direction that the fecal elimination was actually moving at that particular time. So we can have tails that represent different patterns of movement that actually exist as well. Like I say, nothing isn't necessarily straightforward when we're actually talking about the flies. All right, so I've given you an idea that we have more than just insect stains that might be associated with any type of body fluid, but blood in particular. And now we have a rough idea of some of the mechanisms that lead to these stains. Let's do the show and tell piece where I start showing you some of these stains. So what you're about to see first would be fly stains that have been created through interactions with wet blood. The stains themselves are being deposited first on non-porous surfaces, just to give you full context. What we're looking at here then is a relatively representative image of what we would see for regurgitate, so that we see in the center this large, mostly round, circular stain with others that are smaller, much smaller, but similar color appearance. These are various sizes that regurgitate can come in. This is from the same species of flies, not the same individual, but the same species of flies. We're looking at, all the stains we'll be looking at for the most part are from Calliphora vicina, a calophorid, common fly, particularly if we're talking North America, very common if we're talking about cooler temperatures of spring or fall, very common throughout Europe as well. So this is one that will produce larger stains than some of the ones that we would see during peak time during the summer months itself. Next to it is actually a regurgitate stain that contains no blood in it whatsoever, so giving you more of this tan to golden appearance that is important in context when it actually can be used as indication that there was fly activity or a fly has actually fed upon a stain, as opposed to mechanically damaging that stain itself. In contrast, we can get defecatory stains like we see in this particular case that are produced from the passive drop low pressure mechanisms, more or less circular. Defecatory stains show a wider variation in color depending upon the species that we're looking at, from light to very dark, with also having the appearance of the food source that was actually consumed as well. And this gives you context next to that in many cases defecatory stains can be much bigger than regurgitate, but there are other times that that's not the case at all. If the defecatory stain is produced when the fly is actually in motion, as we actually see in this particular case. You wind up getting a stain with the tail that has a tadpole or a sperm cell shape, with the tapering pointing to the left, indicating that's the direction of fly movement. In contrast to that, you can have it where that the fly has created a defecatory stain that does have a small tail that's much less in terms of the length by comparison to the diameter of the stain itself. And in this case, this was a stain that was produced from the fly actually beginning to flight before that it was actually done defecating, hence the smaller tail that we actually see. And there's all kinds of gradations that can be in between. And there are some species that don't ever seem to produce defecatory stains that have a tail at all, making it much more challenging, particularly when those stains have the same morphology as the food source that they fed upon, blood in that particular case. Translocation or transfer patterns are most typically going to take place with the flies when they have walked through wet blood, and then in this particular case drug a portion of the leg, or the other most common is when they drag the abdomen actually through the blood, creating these more or less long lines that may or may not be completely symmetrical or straight. Above that, what you can actually see are these fainter lines, where the same leg was used repeatedly to drag, but there's less and less blood still remaining on the leg to be transferred to a non-bloodied surface. The fourth major type that we come across winds up being tarsal tracks, which are created when the fly walks through the blood or walks over the top of the blood and then deposits footprints in many locations. Now, there is at least one paper that indicates that in their observations, the flies did not have sufficient mass to break the surface tension of the blood drop. But I've worked with the same species and have observed just the opposite. I think it's more of a reflection that the drying state of the blood stain is probably the bigger factor, not the mass of the fly. We do see that tarsal tracks are more likely to be very common at a crime scene when you're dealing with small flies in the genus Chrysomya, Lucilia, and to a lesser extent Formia, and seem to be much less common when we're dealing with large-bodied flies such as Clipper vicina, Clipper vomitoria, Cinnamomya cadaverina, Sarcophaga bullatus. So the tendency that we've seen so far, more testing needs to be done to make sure if that's an absolute observation or not. Just to give you complete contrast, so you're looking at stains that were created with a water-based solution. We did this merely for the video taping that we were doing. We've seen this with blood as well, but we're trying to decrease the contamination of our behavior observation arena that we were using. Two patterns that were quite surprising to us when we actually observed these the first time. One I refer to as purge, where that adult flies actually had recently consumed large volumes of blood and then were repeatedly disturbed by other flies that led to them essentially dropping their payload from the crop or purging the entire contents so that they immediately took flight. Whether I would go so far as to say that's a defensive response, I don't know, but these stains tend to be quite large in diameter. They tend to exceed 4 millimeters, which is not common. Again, they seem to be largely a reflection of purging the entire contents. When you actually test them with presumptive testing or with confirmatory testing, they demonstrate all the characteristics of human blood. So it's a large volume in that case. Cast-off actually is created under different scenarios, but the most common, and actually the one that covered this piece of paper that we're actually looking at, were when there was pooled blood and the flies actually landed in it in such a way that they got wet blood on their wings. Flies do not like to be dirty, and they started to flip their wings and casting off the blood that was on the outside of the wings, that within a 10-second period of time completely started to cover this entire surface. So it's a cast-off appearance that we see on a vertical surface in this particular case, but they do this when the wings wind up having blood on it. They can also fling blood off of the body using their legs as well, because the body's hydrophobic, and there can be droplets of blood that cling under the wings or on various surfaces that the fly actually flings off, creating a cast-off appearance as well. Now, if the surface actually changes so that we're now looking at a porous material, then the characteristics of the stains change radically. So what we're looking at in this particular case are stains that have been produced on a new cotton t-shirt that has not been washed. If I had not been observing the flies, it would be nearly impossible, if not impossible, to distinguish between regurgitate and defecatory stains. Usually tails are not evident in many cases, but what is significant here is that we actually started detecting a new form of defecatory stain, what I reference as fecal spheres, that actually have almost a perfect sphere, three-dimensional appearance to it. There's virtually no absorption and wicking that takes place into the yarns of this fabric whatsoever. And these are defecatory stains that were produced by the forcible elimination from a protruded anus. This suggests that chemically that fecal spheres are distinct from defecatory stains. We have not done the chemical analysis to demonstrate that, but it is significant. And keep in mind, this is a new cotton t-shirt, because by comparison to a t-shirt that has been washed over 30 times, we see that absorption and a small amount of wicking is much more common with these stains, and just very few. In the upper right-hand corner, there's 2 of these fecal spheres by comparison to significantly more when it's an unwashed material. And we see that actually with a number of fabrics, including denim, with various types of carpet as well. And there's still other materials to be tested. In this example, this is dry wick polyester that has not been washed, and we tried to simulate the wear conditions of shirts. So the loose would be someone wearing a baggy shirt versus flat, which is untucked but a regular fit, versus tight, a tucked shirt in that particular case. And what you'll notice is that this was material designed for wicking, and you see substantial increases in wicking of the digestive artifacts when there's tension on the fabric. In fact, a close-up view of that shows you that the parent stain is much darker, and it can be seen in the center, and a secondary stain is the wicking. And this is a relatively low volume. Most of the flies will deposit anywhere between 0.1 microliters to 1 to 2 microliters of liquid in regurgitate or defecate. But there's substantial wicking where that the total stain may represent close to 10 millimeters. Blood behaves in a very similar fashion, so this is something where that the stain appearance and the wicking characteristics are very, very similar with blood, that makes it very difficult to distinguish between them. In contrast, on dry wick polyester, flies that actually are creating tarsal tracks have a very unique appearance, and that there's little wicking, and in fact it looks like that we have these short linear lines from blood that's on the surface of the knit loops in this particular case, most frequently being on the loop legs. There doesn't seem to be any difference between the left or the right loop leg, but the blood is mostly at a superficial or surface level. Now, for some species, we can get sharp contrast so that we can really see the uniqueness in the stains themselves. So what you're seeing here is not from Calliphora vicina. This is actually more typical of 2 flies that we've looked at. Sarcophagus, Sarcophaga bulata, and Cinnamomya cadaverina are the 2 that really showed distinct colored stains. The yellows, the greens, the grays, the browns, and you also get a sense of that dotted line appearance that was created from the fly running as it was actually defecating at the same time. In this scenario, there would be absolutely no difficulty being able to distinguish between what is a true blood stain and what is a fly stain, but that's not the norm for most of the species that we've actually looked at. When we've had situations where we've used new plush carpet, then if I didn't know that these were fly stains, I don't even know that I would suspect that, because it doesn't look like blood. And this is still carpet that most likely has the stain blocker actually on it. It hasn't been swept, it hasn't been washed. And so we see that there's very little absorption and no wicking to speak of that has taken place. We can't distinguish what type of fly artifact. And again, I'm not sure that you would distinguish that it is a fly artifact in that case. It's kind of a similar appearance. This is washed denim. Similar appearance whether we use washed or unwashed denim. In this particular case, that the stains are deposited mostly at a surface level with very little absorption, very little wicking. Very common for this type of material. Many more that we've actually looked at, but we don't have the time to look at all of those. What happens when the fly interacts with dried blood as opposed to wet blood, like what we've just looked at? Well, the morphology is much, much different. So, for example, the stains that we're looking at are on ceramic tiles, so on non-porous surface. In many cases, the stain can be modified. Flaking has taken place here from mechanical interaction. The flies have run over it, they've walked over it, and if you have loose flaking that's taking place and they flap their wings, they can blow that blood stain or portions of the stain a good distance away. When they feed upon it, then you can create perimeter stains, and what you wind up seeing is that they're feeding upon the center. We know that this wasn't due to flaking because we have dried regurgitate that's actually evident right here. In this particular case, it's clear, and we have developed an immunoassay that allows us to distinguish that this was definitely due to fly activity from feeding, so that we can confirm that. Similarly, that you have the formation of a perimeter stain, but in this case, there was such a large volume of regurgitate that was deposited that it wound up having some diffusion of the blood into the regurgitate, staining the regurgitate, even though it was not actually, it was actually the same color that I had just showed you on the ceramic tile. Flies can create skeletal stains so that you just have fragments left over, so this is further consumption. And then in some cases, you can also have skeletal stains that are completely enveloped in the regurgitate, like what we saw with the perimeter stain, as well as having some of the diffusion of blood into that dried regurgitate, still with a skeletal stain itself. If you compare that to dried stains on porous materials, again, we're looking at a new cotton t-shirt. The flies show no interest. This is after 24 hours exposed to several adult flies. We see minimally just one piece of evidence of flaking taking place and no modification of the stain. That is very characteristic of what we see with stains on porous surfaces, whether we use denim like that we're seeing here, whether that we use carpet, whether that we use any type of shirt or other types of clothing apparel. Flies generally show very little interest in dried stains on porous materials itself. I believe it's a tactile issue, and that the mouth parts are sensitive, that any of the fabrics and that the upper topography actually is perceived as being something they don't want to associate with. They will also deposit stains on a corpse, and so I have not had access to human corpses. I've seen pictures that have many fly artifacts on them. In this particular case, replicating that, piglets that we've used in outdoor decomposition, we see that we have many artifacts being deposited on the skin and on the hair. The same is true with guinea pigs in indoor decomposition. The tendency in this case, particularly if we're talking the colder months in the spring or in the fall, the flies tend to go for the head first. Eventually, you will see that the body is totally covered. In the warmer months, we tend to see that there's very little preference to where that the artifacts are deposited, but they are also being deposited on the corpse, whether the skin is fully exposed or whether that the individual is clothed or partially clothed. What does all this mean to us? Well, what we have found from our initial examination of these fly patterns is that there's not nearly as much known about them as we need to know. And I believe they're also undervalued as well. Can they serve as valuable pieces of information in our reconstruction? The answer is possibly, because we also know that there are other features such as time of day when they show preferences for laying this, specific locations on the body, so there might be some time maps that we can derive from understanding the artifacts. More study is needed. We know that the morphologies are much more complicated than most textbooks have referenced. And there has been almost no previous work on porous materials until very recently, and that's mostly out of my research lab. So our complete understanding of the difference of these types of patterns on porous versus non-porous is superficial at best, and we need to know more. We also need to develop much more effective methods for differentiating between bloodstain evidence and fly evidence, and I can tell you that on porous materials, it's virtually impossible to distinguish the two. Does it matter? Everyone's going to have a different opinion about that last statement, but I can tell you that when you put this in the context that fly stains are potential sources of secondary DNA transfer both to and from the crime scene, and that the flies themselves can retain a blood meal for up to 3 weeks and still be depositing artifacts that contain both DNA and protein, and that test positive by any of your chemical tests, Yes, there is very much a need to investigate this further so that we understand and can recognize these types of stains much more effectively than we can now. This was only just a brief introduction. It's an entirely different story to talk about all the methods available for distinguishing these artifacts. So I appreciate your attention, and I'm happy to answer any questions. So feel free to reach out to me if you have any, you want to know more. I'd love to talk to you, but that is the presentation, and I appreciate your attendance. Thank you very much.